Hello everyone, welcome to lesson 1.5 in unit 1, and in this lesson we're going to be going over a detailed account regarding the origin of cells and how cells were even originally created and discovered in the first place. So let's get started. Cell division and the origin of cells. So cells, as we know from the cell theory, can only be formed by the division of pre-existing cells. So each cell in our body was formed when a previously existing cell copied its genetic material and divided into two separate daughter cells. We can trace the origin of our cells in our body to the first cell that was ever created in our body, which is the zygote. So, if we accept that humans evolved from pre existing ancestral species, we can trace the origin of cells back to the earliest cell on Earth. This is where spontaneous generation and the origin of cells can sort of get intertwined within each other. So, spontaneous generation is the formation of living organisms from non living matter. Greek philosophers reported the growth of an organism from seemingly nothing. So, a plant called the siliphium sprouted from soil that was not previously present, and spontaneous generation of mice, frogs, and eels were also reported by certain Greek philosophers from water, air, or decaying matter. So, biologists throughout history remained convinced that spontaneous generation could occur as long as there was access to air. And Louis Pasteur, who was a biologist, decided he was going to end the debate once and for all through a series of experimentation. So in Pasteur's experiment, Louis Pasteur created broths that were stored in swan neck tubes. And the broths were boiled ahead of time before the experiment was conducted to kill any existing and living microorganisms. And Louis Pasteur discovered that growth only occurred in the flask in the form of bacteria if the neck was broken, and thus the broth was exposed to the contaminants from the outside. And this showed that bacterial growth came from external conditions only, and uh, bacterial growth did not occur spontaneously. And here's just a diagram that really emphasizes the findings of the discovery. So you had a broth in a flask that was boiled to kill any pre-existing microorganisms to create a sterile environment. As the broth cooled, condensing water collected, which sealed the mouth of the flask. And in broths where this occurred, no growth of any bacteria, no bacterial growth occurred. And if the neck was broken beforehand, the outside air carried microorganisms into the broth and contaminated the actual broth. And the conclusion is that cells can only arise from pre existing cells and cannot generate spontaneously. So, now with our knowledge that cells can only be formed from the division of pre existing cells, we're going to go over the history of the origin of the first cells on Earth. So, cells were the first living things ever present on Earth. And unless cells arrived on Earth from somewhere else in the universe, they must have arisen from non living material or matter. But this begs the question and inquiry of how a structure so complex as a cell could have arisen by natural means from non living material and matter. And then there are four theories and hypotheses for how some of the main stages could have occurred in the origin of the first cells. So, the first theory is the production of carbon compounds, such as sugars and amino acids. So, Stanley Miller and Harold Urey, which were two scientists, passed steam through a mixture of methane, hydrogen, and ammonia. And electrical discharges were used in this model to stimulate lightning in the environment. And amino acids and other carbon compounds needed for essential for life were produced as a result. The second theory is the assembly of carbon compounds into polymers. So Miller and Urey's experiments allowed for the formation of amino acids, but the conditions used tended to break bonds, preventing polymers from forming. And the first carbon compounds ever present on Earth might have evolved around deep sea vents. And so cracks in the Earth's surface characterized by gushing hot water carrying inorganic chemicals such as iron sulfide. And these chemicals represented readily accessible supplies of energy that could have led to the assembly of carbon compounds that were essential for life. All right, and the third theory was the formation of membranes. So phospholipids or other amphipathic carbon compounds were among the first carbon compounds ever present on Earth. They would have naturally assembled into bilayers. And this is because the formation of the bilayer creates an isolated internal environment that allows for optimal conditions, such as the replication or catalyst to be maintained. All right, and then the fourth theory is the development of a mechanism for inheritance. So, to replicate DNA and pass genes to offspring, enzymes are needed. And to make and produce enzymes, genes are needed. And it is theorized that perhaps RNA was the first genetic material used by cells, as it can store information in the same way as DNA, but it is both self replicating and in itself can act as a catalyst. All right, so now moving into endosymbiosis in eukaryotic cells. So, endosymbiosis refers to the theory that eukaryotic cells may have evolved from prokaryotic cells via endocytosis. This essentially just means that engulfed prokaryotic cells remained undigested by eukaryotic cells when they engulfed them, as it provided new functions to the actual cell. And the theory states that mitochondria were once free living prokaryotic organisms that developed the process of aerobic respiration. It is theorized that larger prokaryotes that could only respire anaerobically took them in by endocytosis and engulfing them and allowed them to live in their cytoplasm as they provided a new function for the actual larger prokaryotic cell. And here's a diagram that really just emphasizes the theory of endosymbiosis and its correlation to present day eukaryotic cells. All right, now moving on to evidence of endosymbiosis. So, mitochondria and chloroplasts have features to suggest they evolved from independent prokaryotes. And this is due to the fact that they have their own genes on a circular DNA molecule. They have their own 70 size ribosomes. They have a double membrane, and their inner membrane has proteins similar to prokaryotes. They are roughly the same size as bacteria and are susceptible to certain antibiotics. They transcribe their DNA and use the mRNA to synthesize some of their own proteins. And they can only be produced by the division of pre existing mitochondria and chloroplasts. And here's just an abbreviation that can help you remember the evidence that suggests that chloroplasts and mitochondria are a result of endosymbiosis. You can remember it through the abbreviation MAD-Doctor. And so for the M membranes, so some organelles have double membranes, and then the outer membrane may be vesicular in origin. The A is antibiotics. Certain organelles like mitochondria and chloroplasts are susceptible to antibiotics. This indicates that the organelles may have bacterial origins. For D is for division. Reproduction occurs via a fission-like process, similar to prokaryotic cells. The other D is for DNA. Mitochondria and chloroplasts have their own DNA, which is naked and circular, like prokaryotic DNA structure. And then finally, the R is ribosomes. Mitochondria and chloroplasts have their own ribosomes, which are 70S in size, which is identical to prokaryotic ribosomes. So that's it for lesson 1.5, the origin of the cell. Thank you for listening, and I'll see you next time.